Hi everyone, and welcome to our summary on protein structure and bonding. So in our previous video we had a little look at amino acids, but what we're going to do in these next couple of videos is look at proteins in more detail themselves. So the spec statement that we're going to be focusing on in this video is looking at those levels of protein structure and the bonds that are involved within them. The first level of protein structure then is the primary structure. If you are ever asked on an exam paper, what is the primary structure of a protein? All they're looking for is the sequence of amino acids, because that's literally what the primary structure is. It just tells us in what order those amino acids occur in. So does it go proline, cysteine, lysine, etc.? That's the primary structure, just this simple sequence of amino acids. The sequence that those amino acids occur in, and therefore the primary structure, is going to have a part to play in these other levels of protein structure, because those individual amino acids all have their own little side groups, remember, and they all cause things to interact in different ways, as we'll find out as we go through this video. So basically this order of amino acids in the primary structure is going to play a part in determining that final shape of the protein molecule. So primary structure, sequence of amino acids. The secondary structure, our second level here, this is either the coiling or folding of our polypeptide chain. So a single polypeptide chain, it's looking to see what's happening to it. Is it going to be coiling into what we call an alpha helix, or is it going to form a beta pleated sheet? So your secondary structure, coiling or folding into either an alpha helix or a beta pleated sheet. Let's have a little look at the alpha helix in a bit more detail then. We can see a little diagram of it on the left hand side, or at least a section of the alpha helix there. And what we find is that each 10 turns of our alpha helix, of that coil, is actually going to contain 36 amino acids. So 3.6 amino acids per turn. Far easier just to think of it as 36 per 10 turns of the coil. Now, we've said it's going to coil, but it's not just going to sit there because it feels like it. It's going to have to be held in shape by a certain type of bond. And we're coming back to our good old friend, the hydrogen bond. Now we can see these in a little diagram by the little dotty lines between the NH, okay, so between our nitrogen joined to a hydrogen there, and the carbon double bonded to the oxygen. So we've obviously got one end of an amino acid, basically what was the amino group, and then what was the carboxyl group of a different amino acid. So what we can see is we get this hydrogen bond forming between the oxygen and the hydrogen there, and that's going to occur on the carbon oxygen group of the amino acid that is 4 places ahead in the chain. So if this is our amino acid, here we go, if that's number 1, then this one here where the bond actually is would be number 5, because it's 4 places ahead. So we end up with these little hydrogen bonds between those 2 parts of the amino acids, and that's what holds it in that coiled shape, the alpha helix. The other option is a beta pleated sheet. Now, the beta pleated sheet is basically formed when our polypeptide chain folds itself into like a zigzag structure. And you can see a little diagram at the bottom there. So what we end up with is this little kind of like zigzaggy bit here, and then it folds back and we get another little zigzaggy bit here, and so on. Okay, now it's again held in that same shape by hydrogen bonds. So between the NH of one amino acid and the CO of another further down the strand. So if we look at our little diagram here, what we can see is here's our little NH group, and there's our CO, and between it we've got our hydrogen bond. So what we have, hydrogen bonds then occurring between these two sections, holding them in that beta pleated sheet. So secondary structure, alpha helix or beta pleated sheet. One thing to bear in mind when we're talking about these secondary structures of the alpha helix and the beta pleated sheet is that when they're at optimum temperature and optimum pH, they're actually really stable as a structure. And that all comes down to the large number of the hydrogen bonds that occur within those structures themselves. Now, we don't always have a situation where our polypeptide chain only has an alpha helix in it, or only has beta pleated sheets. There are some of these polypeptide chains that will have alpha helix at one part of the chain and then the beta pleated sheet at a completely different region of the chain. So it's not just either alpha helix or beta pleated sheet. Some of these polypeptide chains have a mixture of the two. So secondary structure, alpha helix or beta pleated sheet forming within that individual polypeptide chain. Next up is the tertiary structure. Now, tertiary structure, quite simply, is the three-dimensional shape of the molecule. So tertiary, 3D. Tertiary obviously means 3, so we are talking 3D shape in the tertiary structure. Nice easy link there, I hope. Now, what we find is that the whole reason it stays in that three-dimensional shape, which is very specific, is because of the bonding that occurs between those side chains on the amino acids. So remember what we said, we've got our little amino acid, so let's just quickly sketch it out again as a reminder. And this is the key bit here. What that R group is, that side chain, is going to determine what kind of bonding is going to occur between one amino acid and another. So what we find is, depending on what amino acids are present in that primary structure and the sequence of them, then we'll get different shapes in this 3D structure here, this tertiary structure. They can form a supercoiled shape, and these are what we find in our fibrous proteins, or they'll make a spherical shape. And this is what we see within the globular proteins. And we'll look at fibrous and globular proteins in more detail in another video. The last level of our protein structure then is the quaternary structure. Now, this isn't going to be present in all of our proteins. The quaternary structure will only be present in those that have more than one polypeptide chain present. What the quaternary structure does is it describes how those different chains are arranged to give the final structure of the protein. So all proteins have primary, secondary and tertiary structures, and then some, those that are made of more than one polypeptide chain, have quaternary structure as well. We did briefly mention bonding. What we're going to do now is go through the different types of bonding that can occur within our proteins in a bit more detail and see where they occur and what it actually means for our proteins. So, nice little recap on the different bonds that we find at the different levels of protein structure, because this does come up from time to time on your exam papers as a question. First of all then, primary structure. Remember, your primary structure is the sequence of amino acids. So we're looking at a single chain of amino acids. That is the peptide bond, because between each amino acid we find peptide bonds holding them together. So primary, peptide. Secondary structure, alpha helix or beta pleated sheet. This is all held by hydrogen bonds. Then when we come up to the tertiary and quaternary structures, this is going to be hydrogen bonds again, but also other types of bonding. And the other types of bonding, I know it sounds like I'm being just really quite vague at this point. That is deliberate. It's not just that I don't know what it is. That all because it comes down to what amino acids are actually present. Because depending on what amino acids we have present, that determines what kind of bonding we will see within our tertiary and quaternary structures. Let's have a bit of a closer look at these key bonds then. First one, hydrogen bonds. We keep coming back to these in our biological molecules topic. If you don't remember the word hydrogen bonds, then I'm going to be very concerned about how your A-level is going to go. So, hydrogen bonds form between a delta positive hydrogen and another atom with that delta negative charge. So what we find is they're going to be forming between hydroxyl, carboxyl and amino groups, okay? So we've got slight charges on a hydrogen, positive ones obviously, and a slight charge of a negative form on something else. We're a little bit more flexible in the something else. If we've got a polar R group, so basically if that side chain is polar, then we can get those hydrogen bonds forming between that R group and an oppositely charged R group on a different amino acid. That's our hydrogen bond. It is worth remembering, as we've said previously, hydrogen bonds are weak, and in fact of the bonds we're going to be looking at in fast protein structures, they are the weakest of the bonds. But if we're looking to see the effect of having lots of hydrogen bonds within a structure, it makes it strong. Because if you've only got a single hydrogen bond, that bond is weak, it's easily overcome. If we've got many hydrogen bonds, then we have a strong structure because it's going to take more energy to then overcome all of those hydrogen bonds. So do remember, hydrogen bonds are the weakest, forming between a delta positive hydrogen and a delta negative atom of another kind. Next up, we've got the ionic bonds. Now, we find these forming between carboxyl and amino groups that are part of R groups. So what has to happen first of all is that they need to become ionized. So ionized means they're going to become charged. So our carboxyl group is going to lose the hydrogen to become COO-, and our amino group is going to gain the hydrogen to become NH3+. So we now have these charged groups. Because they've got opposite charges, one is negative, one is positive, then they will attract through this strong electrostatic force of attraction. And that is an ionic bond. Same thing we said in GCSE chemistry, a strong electrostatic force of attraction between a negative and a positive ion gives us the ionic bond there. And these are the strongest of the bonds we are going to look at. Hydrogen bonds the weakest, ionic bonds the strongest. The next type of bond we're going to look at are disulfide links. These also, depending on what you are reading, go by the name of disulfide bridges, disulfide bonds, disulfide links. They have an array of names, but they all have the one thing in common, disulfide. Now, what we find is if we look at all of the R groups of the amino acids and we find cysteine, then you will see that that R group contains a sulfur atom. Now, disulfide. Di means two, sulfide referring to sulfur atoms. So we've got two sulfur atoms. That's what's making this bond, this link, this bridge, whatever you want to refer to it as. So what we actually find is these disulfide links are actually strong covalent bonds forming between the two R groups of two cysteines. So we've got two cysteines, and then because their R group consists of a sulfur atom in each case, we then form a covalent bond between those two sulfur atoms, and that is the disulfide link, disulfide bridge. The last one to bear in mind are hydrophobic and hydrophilic interactions. As we mentioned previously, those R groups could be hydrophobic, therefore water hating, they don't really want to be near water, or they can be hydrophilic, water loving, they want to be near the water. What we find is, depending on whether they're hydrophobic or hydrophilic, that's going to influence how they arrange themselves in that structure of the protein. If they're hydrophobic, then they're basically going to orientate themselves towards the center of the polypeptide. So basically making sure they're as far away from the water as they can get. If they're hydrophilic, these bits tend to be pointing outwards around the edge of our polypeptide towards the watery environment. Because we're seeing those two different actions, hydrophobic turning inwards, hydrophilic turning outwards, we end up with this twisting of the amino acid chain. So again, that's contributing to that final shape of the molecule. When it comes to these bonds, do make sure that you can put them in order of their strength. So when we're looking at that, obviously, if you know your ionic bonds are the strongest and your hydrogen bonds are the weakest, whatever other bond they give you just goes in the middle. They're normally only going to give you 3 in this, so if you know the 2 ends, middle bit's just easy to fill in from the others. As always, I do hope you found this video useful. Don't forget to subscribe so you can see when we upload more videos, and of course there are some additional resources over on my website as well that hopefully will help you out with your A-level biology.